Oh, good. Where do you get them? I had them?
Yeah. Where they at? Because that's what I'm wondering. Where they at?
Oh, I put, you took them here. I got them. I put it in my overhead stack.
Okay.
I'm trying to keep myself.
Okay. Okay, but the next witness is
Brian.
Okay.
All right, go to back in session.
Bring the jury back in. Get the next witness.
I do, Judge. Can I let the last one go? I don't, before somebody else. Thank you, Judge.
You may be seated.
Yes, Judge, they are.
Yes, Charlie.
Judge, people call it Brian Schloss.
Very well.
Yes.
You may proceed.
Thank you, Judge. Good afternoon, sir. Could you please state your name and introduce yourself to the jury?
My name is Brian Schloff. I'm a forensic scientist with the Michigan State Police Crime Lab in Northville, Michigan,
And how long have you been a forensic scientist?
since April of 2009.
And in order to be a forensic scientist, do you have any special training or experience?
Yes, I was trained by the NFSTC, which is the National Forensic Science and Technology Center, in conjunction with the Michigan State Police as a forensic scientist in the biology unit
And when did you receive that training?
in 2009.
And prior to that, did you have any higher level education that you attended?
Yes, I have a Bachelor of Science degree from Michigan State University in zoology with a concentration in genetics. I also obtained some graduate coursework at Michigan State University as well as at the University of Florida, where I obtained a certificate in DNA analysis and serology.
And do you have any formal education in DNA analysis?
Yes.
And what is that education?
My formal education in DNA analysis is my bachelor's degree from Michigan State University as well as my graduate coursework.
Okay. And does it also include the training that you were talking about as a forensic scientist?
Yes.
Okay. And does that education include hands-on work with DNA and science?
Yes.
And have you done any special research or published any papers on DNA analysis?
I do perform or participate rather in continuing education. I also perform proficiency tests twice annually, and I have been trained in technologies relating to DNA analysis.
Did you have any special training or certifications?
The specialized training that I receive is in the area of DNA analysis. I do also attend conferences, DNA conferences, as well as participate in biology unit training events conducted by the Michigan State Police.
And what about certifications? Are you certified to work in a particular area?
I don't have certifications. DNA analysts aren't certified in the state of Michigan, but our laboratory is accredited by the ASCII lab.
Okay. And by whom?
By the American Society of Clinical Laboratory Directors Laboratory Accreditation Board.
Okay, thank you. So at this time, I would ask to qualify Mr. Schloss in DNA analysis and body fluid identification.
Brief response.
You may voir dire.
Judge, I don't believe it's necessary for me to voir dire at this time. I have had the opportunity to look over this gentleman's credentials, including his curriculum vitae or CV, and we have no issue with him being qualified as an expert.
Very well. Witness may testify as an expert in that field.
You already mentioned that you are a forensic scientist, and I'm not sure if you mentioned, are you a laboratory manager as well?
Yes, I am.
Did you mention that and I forgot?
No, I didn't mention that.
Okay.
I am a laboratory manager at the biology unit at the crime lab.
And what is it that you do as a laboratory manager?
I manage a team of forensic scientists whose job is to analyze biological evidence obtained from crime scenes for the presence of body fluids or DNA, and if found, make comparisons to known reference samples that are submitted, and then report conclusions.
So I want to direct your attention specifically to lab number NZ162547. Did you have occasion to do what you just described in this case?
In this case, I received data that was generated, DNA profiles that were generated, and I compared those DNA profiles to known reference DNA profiles that were submitted.
And did you receive the samples from another forensic scientist?
Yes, I did.
And was that Sarah Barr?
Yes.
Okay. And so Ms. Barr did not make any type of comparisons, meaning she didn't try to determine whose DNA it was that was on those samples. Is that true?
That's correct.
Okay. But that was what you did in this case?
I made the comparisons and reported the conclusions for those comparisons.
Okay. And when you reported the conclusions, did you put all of your findings in a report?
Yes.
And is that common and part of your general practice to put everything that you did into a report?
Yes.
And is that report given the same lab number that those samples are given and that we've just talked about?
Yes.
Judge, may I approach?
You may.
I'm going to show you some pre-marked identification to go proposed exhibit 165. Can you take a look at that? Did you recognize that?
Yes.
And what is that?
This is the report that I offered.
And does that report contain your conclusions that you made in this case?
Yes.
This is the time I've moved to exhibit 165.
No objection, Judge.
Shall be received.
All right. What type of items did you receive in this case?
Can I refer to my report?
You may. Will that refresh your recollection?
Yes.
You may.
In this case, I received 5 evidence DNA profiles and 2 known reference DNA profiles. The 2 reference DNA profiles, one is for Natalia Muntean and the other is for Radu Muntean.
Okay, and can you explain briefly what a known reference sample is?
Sure. In this case, the report reflects that they were buccal swabs. A buccal swab is it's kind of like a Q-tip or a cotton swab, but it's sterile. And it is a specialized swab that's used to collect cells from the inside lining of a cheek. And those cells can be used because they contain DNA, and they can use to develop a DNA profile. They can be used as a known reference for that person who they're collected from.
And so you said 2 known reference samples, and then you mentioned other evidence samples. What were those samples?
Yes. I received DNA profiles from the following evidence items: possible blood staining from a roll of duct tape, possible blood staining from apparent hairs, possible blood staining from right front door handle, possible blood staining from seat cushion, center console, and possible blood staining from jacket.
Okay, and let's start at the top with the possible blood staining from the roll of duct tape. So you received a DNA sample from that possible blood staining on the roll of duct tape. Is that right?
Yes.
Okay. And what did you find with regards to that DNA sample?
I found that it was consistent with a mixture of 2 individuals, and then I made comparisons to the known reference samples, and then I reported the conclusions in the form of likelihood ratios.
So to kind of break it down, so you found in the sample that you received, there was DNA, and it looked like it came from 2 people. Is that right?
I received the DNA profile from that evidence item, and it does, and I did conclude that it contains a mixture of DNA types, meaning that more than one person contributed to the DNA sample, and that it's consistent with 2 individuals.
Okay. And then you looked at the reference samples that you were just telling us about, so the samples, we know whose DNA that is, in this case, Natalia Muntean and Radu Muntean. Is that right?
Yes.
You compared the DNA sample that was the mixture of the 2 to those known samples.
Yes, I compared them through a probabilistic genotyping system to determine whether or not it was likely that either of them could have been a contributor to the DNA.
Okay. And you mentioned the term likelihood ratio. What is a likelihood ratio?
A likelihood ratio is a mathematical way of comparing two propositions or ideas. It is, generally speaking, it's constructed to determine whether or not a person of interest could be a possible contributor to the DNA that's found in the evidence.
Okay. And so with regard to the DNA mixture on the roll of duct tape, what did you find as far as the likelihood ratio?
Sure. I set up two different likelihood ratios for this particular sample to compare to both of the reference samples that were submitted. The first likelihood ratio that I constructed was to determine whether or not Natalia Muntean was a possible contributor to this DNA sample. So the ideas that I'm comparing in this case are, is this, the question that I'm asking is this actually does this mixture contain Natalia Muntean and one unknown contributor, or is this a mixture of two other unknown contributors? And in this case, based on those results, it is at least 190 septillion times more likely that it originated from Natalia Muntean and some other contributor rather than from two unknown contributors.
Can you kind of break that down for us in layman's terms and tell us what that number means and kind of what that result means?
Sure. It means that there is very strong support that Natalia Muntean is a contributor to the DNA mixture.
Do you know how many zeros are in a septillion?
Yes.
How many zeros?
24 zeros.
Okay. So that's a really big number.
Yes, it's very, very large.
Okay. Did you reach any other conclusions as to the likelihood ratio with respect to the possible blood stains in the roll of duct tape as to the second contributor?
Yes. The second likelihood ratio that I constructed was to determine whether or not Radu Muntean was a contributor to this DNA mixture.
And what did you find?
Found that, well, the results indicate